Hello everyone, I am Dr. Shilpa Hindlekar, Assistant Professor at Government College of Arts, Science and Commerce, Kepe. Welcome to the program of Bachelor of Science, third year. The subject is Botany, semester 6 and the course code is BOC 110 and the course title is Plant Ecology and Phytogeography. The title of the unit is Plant Communities and the module name is Characteristics of Plant Communities 2. The outline. It includes the analytical characters of plant community, wherein we are going to study the qualitative and quantitative characters of plant community. Learning outcomes. At the end of this module, the student will be able to describe the qualitative characteristics of different plant communities and calculate the quantitative characters of plant community. The characters used in a plant community. These characters are broadly classified into two major categories. The first one is the analytical characters and the second one is the synthetic characters. The analytical characters are expressed in terms of 5-point scale and the synthetic characters are actually computed from the analytical characters. Now, analytical characters again are subdivided into quantitative characters and qualitative characters. Quantitative characters include frequency, density, abundance, cover and basal area. Qualitative characters include physiognomy, phenology, stratification, abundance, sociability or gregarious, vitality and vigor, and life forms which are also called as growth forms. Coming to the first quantitative character, frequency. What is frequency is the number of sampling units in terms of percentage in which a particular species occur. The various species of a community are recorded by different phytosociological methods by using any sampling units like quadrats, transects, or point methods. The frequency of each species is calculated as follows. Frequency in terms of percentage is equal to the number of quadrats in which a species occurred divided by the total number of quadrats studied into 100. After determining the percentage frequency values of each of the species, they are then distributed into 5 frequency classes of Ronquier from 1934. These are the classes where it is distributed, wherein if the frequency is from 0 to 20%, it comes under frequency class A, 21 to 40%, class B, 41 to 60%, class C, and 61 to 80% D, and last 81 to 100% is the highest class, the E. Second is density. Density represents the numerical strength of a species in a community. The number of individuals of a species in any unit area is its density. Density is calculated wherein the total number of individuals of a species in all the quadrats divided by the total number of quadrats studied. The value thus obtained is then expressed as the number of individuals per unit area. Density values would give you an idea of competition that is occurring among the species. Third is abundance. It is the number of individuals of any species per sampling unit of occurrence. It is calculated as follows. Abundance is equal to the total number of individuals of the species in all quadrats divided by the number of quadrats in which the species occurred. The fourth is cover. Cover is the area that is occupied by the plant. This cover may be the herbage cover or the cover or just the basal area. So cover or herbage is the area or ground that is occupied by the above ground parts of the plants like leaves, stem, inflorescence as viewed from above. If this area is demarcated by vertical projections, the area of the ground covered by the plant canopy, it is called as a canopy cover or it is called as foliage cover or herbage cover. This is estimated by using chart, line intercept or a point frame method. Basal area refers to the ground that is actually penetrated by the stems. It is measured either 2.5 centimeters above ground or on the ground level. Here it is measured by line interception or point centered quadrat method. Now based on this cover values, the species are again grouped into 6 groups. Group 1 is less than 5%, Group 2 is 5 to 25%, Group 3 is 25 to 50%, Group 4 is 50 to 75%, Group 5 is 75 to 90%, Group 6 is 95 to 100%. Here this diagram would show you the herbage cover, when relationship between the herbage cover and the basal area in different types of growth forms, wherein you have the herbs So this is the basal area here. In shrubs, this is the area which is demarcated, and in trees, this is the area. The qualitative characters include physiognomy, phenology, stratification, abundance, sociability or gregarious. Sixth is vitality, and the last is life form and growth forms, which will be discussed in the next module. Now, most of them are grouped according to the point scales. Physiognomy is nothing but it is the general appearance of vegetation as determined by the growth of the dominant species. So, it can be expressed by a single term, which may be, for example, forest. If the dominant vegetation is in terms of the tropical forest, grasslands when you have the the grasses as the dominant species, and deserts you'll have the desert plants as the dominant species. So based on the type of vegetation that is appearing, you would classify them as grassland forest or the desert forest and so on. Phenology is the scientific study of seasonal changes, that is the periodic phenomena of organisms in relation to their climate. Different species have different periods of seed germination, vegetative growth, flowering and fruiting, leaf fall, seed and fruit dispersal. Now, such data of individual species are recorded in phenology. A study of the date and the time of these events is called as phenology. So, phenology is a calendar of events in the life history of the plant, which is shown by means of phenograms. Here you can see in this picture a phenogram, wherein it shows the germination as 1, the second is vegetative growth, third is flowering, then fourth is fruit formation, fifth is seed maturation, and sixth is the death of the plant. Where this particular data would show you the phenogram of different species occurring, wherein in March here the death of the plant occurs of Digitaria royalina. Then you can see vegetative growth occurring here in July and flowering also occurring. Then you have in August again it should show flowering, fruit formation, seed maturation and so on. And as you go towards the October, it is showing the death of the organisms. So these are the phenograms of some of the grasses and sedges of the Indian grasslands. Stratification, the way in which different species are arranged in different vertical layers in order to make full use of the available physical and physiological requirements. The data here is obtained by using a bisect. The vertical projection is constructed and the stature of the species may be plotted to find out the distribution pattern of stems, roots, etc. of different species. Abundance, plants are not only uniformly distributed in an area, they are found in small patches or in groups differing in number at each place. So it is divided into 5 arbitrary groups. Those species which are very, very rare, then those which are rare, third one is common, the one which are occurring frequently, and those which are very much frequent in a particular area. Sociability, it denotes the proximity of plants to one another. Plants generally grow as isolated individuals in patches, colonies or groups. The plants of some species may grow better when they are nearer to each other and produce a thick population. Others may become weak or they die in such associations. The species with same density may differ in sociability values. Thus, it expresses the degree of association. Sociability is again divided into 5 classes: S1, S2, S3, S4 and S5. S1 is the plants which are found separately from each other. S2, wherein you find a group of 4 to 6 plants. S3, many small scattered groups. S4 is several bigger groups of many plants at one place. And S5 is a large group occupying a very large area. Vitality, it is the capacity of normal growth and reproduction. It depends on the weight of the species. In plants, you have the stem height, the root length, leaf area, leaf number and weight of the flowers, fruit and seeds, which would determine the vitality. Now, Daubenmeier in the year 1968 grouped vitality into 5 categories: V1, V2, V3, V4 and V5. V1 are the plants whose seedlings would die. V2, seedlings grow but they are unable to reproduce. V3 is they would reproduce only vegetatively, and V4 is reproduce sexually but they are uncommon. And V5 wherein they would reproduce sexually and grow regularly. These are the references which I have used for the preparation of this e-content. Thank you.